Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and today we are going to start the new module, and the new module is going to deal with the molecular biology tools. So, the molecular biology tools normally deals with the molecules which are important for the livelihood or for the maintenance of the life. So, there are four major molecules which are important for maintaining the life. One is DNA, the second is RNA, the third is protein, and the fourth is the lipids. So, molecular biology normally deals with all the macromolecule, especially the DNA, RNA and proteins. So, when we talk about the DNA is being amplified with a technique known as the polymerase chain reactions. So, as the name suggests, the polymerase chain reaction is a technique which is used to amplify a lot of double stranded DNA molecules with the same identical size and the sequence by the enzymatic method and the cyclic conditions. What it means is that you have original DNA, which is the original DNA, and this original DNA is being amplified for a multiple cycles, and that is how you are actually going to have the multiple copies, like you have the 4 multiple copies which are actually going to be identical, which means the sequence of all these fragments are going to be identical to the original sequence, and also they are going to be amplified with the help of the enzymes. So, if you want to understand the process of polymerase chain reactions, you have to understand the two basic component. One, you have to understand about the DNA. The second, you have to understand about the enzymatic machinery, what is important for amplifying the DNA. So, let us start with understanding the DNA first, and then we are going to understand about the machinery, and then subsequently we are also going to understand how the people have developed the technique, and then we are also going to understand all the technical aspects related to polymerase chain reactions. So, DNA is a nucleic acid, and that is composed of the two complementary nucleotide binding blocks chains, which means in a typical DNA you have the two chains, one is the 5 prime to 3 prime and the 3 prime to 5 prime. Both are these strands are connected by the nucleotides what is present inside, and the nucleotide are made up of a phosphate group, a 5 carbon sugar and a nitrogen base. So, as you know that when the base is adding up with the sugar, it is actually forming the nucleoside, and when the nucleoside is being attached with the phosphate, it is called as nucleotide. So, the DNA is made up of the nucleotides. So, it is actually a macromolecule where you have the one chain of the nucleotides connected to the another chain of nucleotides, and these two chain of the nucleotides are complementary to each other. So, DNA has the 4 nitrogen bases. You have the 2 purines like the 2 ring bases like adenine and guanine, and then you have the 2 pyrimidines like cytosine and the thymine, and these 4 bases are linked in a repeated pattern by a hydrogen bonding between the nitrogen bases. The linking of the 2 complementary strand is called as the hybridizations. So, A is making a pair with T and G is always making a pair with C. So, what you see is that the A is making a pair with T with the help of the 2 hydrogen bonding, whereas the G is making a hydrogen bonding with C with the help of the 3 hydrogen bonding, which means the strength of the A to T interaction is weaker compared to the interaction of G to C. and because the A is making a pair with T and G is making a pair with C, the DNA is complementary in nature. What is mean by complementary in nature is that if you have a first sequence which is called as the primary strand, then you can be able to deduce the complementary sequence. For example, in this case we have a primary sequence which is GGC, TAT, GTG, and what you see is that wherever you have the G is actually having a C at the complementary strands. So, that is the way it is actually going to maintain the complementarity. How the complementarity is going to help you in terms of the conserving the molecules, because if you have the strand 1, you can be able to generate the strand 2 because the strand 1 is complementary to each other, or if you have the strand 2 you can be able to generate the strand 1, and that is the basic principle through which the PCR mechanism is working. For example, if I have this particular sequence and if I want to synthesize the strand 2, what I can do is I can simply attach a short nucleotide bases and then I can just put the machinery, and that machinery is actually going to synthesize the complementary sequences, which means I am looking for a machinery which actually can recognize this DNA sequence, and then that machinery could be able to understand that the DNA is complementary in nature. So, what will happen is if I have this sequence, the machinery is going to sit onto this sequence, and then it is actually going to accept the nucleotide from the what you are going to supply, and then it is actually going to synthesize the complementary strand, and once it reaches to the end of the sequence, it will know that now the synthesis is over. So, it is actually going to terminate. So, now so far what we have discussed about the molecule which is actually going to be amplified during the polymerase chain reactions, and now what we are going to understand? We are going to understand about the machinery. So, the DNA synthesis machinery is the basis of the life on the earth, or the DNA synthesis machinery is very crucial for duplicating the DNA, and DNA synthesis is linked to the cell division as well as the growth. So, this is an example of the bacteria. So, what you see is that we have the bacteria which is actually having a single genome copy, and what will happen is that the first event itself is that the genome is actually going to duplicate into 2 copies. So, ultimately it is going to have 2 copies of the genome, and then it is actually going to be divided longitudinally, and that is how you are actually going to have the 2 bacterial colonies. Similarly, for in the prokaryote, it has the multiple events through which it actually goes through, and then it is actually going to be divided into 2, and that all events are actually being called as the cell cycle. What are these events? You have a G1. So, in the G1 you are actually going to increase the amount of cytosol, and you are actually going to prepare the cell for the synthesis phase, and then you are actually entering into a synthesis phase. So, in the synthesis phase or the S phase, you are actually going to do a DNA synthesis, which means you are going to make a first copy of DNA and you are going to synthesize the second copy of DNA, which means now the DNA is going to be duplicated, which means now you are going to have the 2 genomes just like in this stage, and now it will enter into the G2 phase. G2 is also a preparative stage. So, here also there will be some additional preparative stages, and then the cell will enter into the mitotic phase, and in this phase the cell is going to divide into 2. So, the original cell will again go through with the cell cycle, whereas it is actually going to give you the one more copy of the cell. So, let us say that if you actually block the DNA synthesis, if you block this step, you are actually going to disrupt the cell cycle, and that is how you are actually going to stop the multiplication as well as the growth of particular cell. So, process of duplication of the entire genome prior to the cell division. So, DNA replication is a process which occurs in the cell prior to the duplication of the cell. So, and that occurs within the S phase. So, it has a biological significance that the DNA replication is a very accurate process, and it requires to in order to preserve the integrity of the genome in a successive generation, which means if you are actually going to generate the mutations, or if you are start generating the modulations within the sequence of the genome what you got from the original cell, you are eventually going to keep altering the DNA sequences, and that actually is not going to carry the same information, because the major function of the DNA is to carry the genetic information, so that it is actually going to allow the daughter cells to keep running the similar kind of metabolism. So, that is why it is important that the DNA replication should be accurate, so that it should produce the identical copy in order to preserve the sequence of the DNA, as well as it should preserve the integrity of the genome, and that should continue for several generations. In eukaryotes, the replication only occurs during the S phase of the cell cycle. The replication rate in the eukaryote is slower, resulting in a higher fidelity accuracy of replication in eukaryotes. Now, the DNA replication is having the various characteristics. For example, DNA replication is semi-conservative, which means the DNA replication is going to be semi-conservative, means when you are actually going to synthesize, for example, if this is the 2 strand 1 and 2, it is actually going to give you the 4 copies of 4 strands, right. But in this 4 strand, the 1 and 2 are going to be separated, and the 3 and 4 are actually going to be synthesized. So, the new strand what is being synthesized is actually going to make a pair with the original strands, and that is this process is called as the semi conservative modes. So, this is the 1 and 2 are actually coming from the parents, whereas the 3 and 4 are actually the newly synthesized strands. This means the information what you have in the original DNA is going to be divided into 2, and that is why it is not going to be conservative, which means it is not going to make the pair like 1 2 and 3 4, it is actually going to make a pair like 1 and 3 and 2 and 4. So, that is actually a semi conservative mode of replications. If you want to study more about, or if you are interested to study more about how people have discovered this semi conservative mode, you can easily go through some of the molecular biology book, and you can read about the some of the classical experiments what is being designed, and how people have discovered that the DNA replication is semi conservative in nature. It starts at the origin of the replications. So, DNA synthesis does not start at random places, it requires a place where it actually can start. So, that place is called as the origin of replications. In the bacterial cell you have the single origin of replications, whereas in the eukaryotic cell you have the multiple places of the origin of replications, because the bacterial genome is smaller in nature. So, it actually does not require the multiple original replications. Compared to that, the eukaryotic genomes are bigger in size. So, they require the multiple origin of replication, so that the replication can start at multiple places, and that is how it actually can be able to complete the replications in a given time frame, because you know that the replication is important for making a second copy of the genome, so that the cell will enter into the G2 and M phase, and that is how it actually going to divide and give you the more number of cells. So, that is why the and S phase is actually the bottleneck of the whole process, and that is how the DNA replication has to be in synchronous with the mitotic as well as the G2 phase. DNA synthesis by the DNA polymerase always occurs in the direction which is called as the 5 prime to 3 prime directions. So, you know that the DNA is actually a polymer of the nucleotides, and nucleotides have 2 strands like 5 prime and the 3 prime. So, if I draw a typical nucleotide structures, what you will see is that this is actually a sugar, then this is the base. So, for example, I have put a and here you have the sugars, and on this fourth carbon you have the fifth carbon, and then you have the CH2, and then you have the phosphate group. So, this is actually the 5 prime end and this is the 3 prime end, because here you have the OH, this is the 3 prime end and this is the 2 prime end. So, this is actually you are going to have an OH, and that is OH what you can actually be able to use. So, if a DNA synthesis is occurring, it is actually either utilizing this 5 prime OH or the 3 prime OH, which means the that is why the DNA has a directionality which actually goes from 5 prime to 3 prime and 3 prime to 5 prime, because this is the complementary strand. So, the DNA synthesis always occurs in a direction which is called as the 5 prime to 3 prime, which means it actually going to start the synthesis on this strand, the strand which is having the 3 prime ends, and that is how it is actually going to synthesize in this direction, because the synthesis will be in this direction. It could be unidirectional or the bidirectional, which means it can be either in this direction or it can be in both the directions simultaneously. It is a semi discontinuous, for example. So, semi discontinuous means it is actually going to continue for some time, and then it is actually going to stop, and then it is again going to continue for that, and that is why it is actually going to create the two different types of strands. One is called as the leading strand, the other one is called as the lagging strand. So, this is the strand which is actually synthesizing the DNA in the multiple steps is called as the lagging strand, whereas the strand which starts from the one end and continues like that is called as the leading strand. And for any DNA replication process, you require a primer, and that primer is being synthesized by an enzyme which is called as the RNA primase, and the primers which are being used in the DNA replication are made up of the RNA, and this is being synthesized by the RNA polymerase. DNA replication has 3 steps. It has a like initiations. So, in the initiation the DNA is going to be prepared for the synthesis. What is mean by the initiation is that in the initiation step the protein will bind to the DNA. So, you know the DNA is a double helical structure. So, this double helical structure first has to be straight. So, that first you have to remove the helicity of this particular DNA. So, you are going to first generate the DNA like double stranded DNA, and then you have to actually break the two strands separately, so that it is actually going to be ready for accepting the synthesis machinery, and that is what the events are happening when you are actually going for the initiation states. So, in the initiation state the protein will bind to the DNA, and then there will be a opening of double helix DNA, so that it is actually going to be single stranded DNA. So, the single stranded DNA is going to be generated in the initiation stage. Then you have the elongation steps. So, in the elongation steps the DNA machinery is going to be sit onto this and as well as onto this, and then it is actually going to start adding the nucleotides, and that is how it is actually going to start the synthesis of the second strands. So, in the elongation steps the protein will connect the correct sequence of the nucleotide into a continuous new strands of DNA, and once the DNA machinery is going to be reached to the corners like to the end of the sequence, it is actually going to stop the synthesis, and that is what the third step, that is third step is the termination, which is actually going to stop the DNA synthesis. And once the DNA synthesis is over, the DNA has to again regain to its original form, and that is the step what is required before you do the termination, which means once the synthesis is over, the DNA is first going to be converted into a double helical structure, and then from double stranded structure you have to generate a double helical structure. So, that is why DNA synthesis is a very fine controlled process, where you first have to do the initiation, where the DNA has to be break into the two different strands, and then the two different strands are going to be occupied by the DNA synthesis machinery. And what the DNA synthesis machinery is going to do? So, DNA synthesis machinery involves the DNA polymerases, which actually going to sit onto the enzyme, and then they are actually going to read the original sequence, and based on the original sequence they are actually going to add the nucleotides onto the second strand, and that is how it is actually going to start the synthesis of the second strand. And once they reach to the, and that is what the elongation phase, and once they reach to the end of the sequence, they are actually going to start the termination steps. So, once the DNA synthesis is over, it is actually going to generate the double stranded DNA, and then double stranded DNA is actually going to be coiled, and then it is going to generate the helical structures. There are different enzymes what is involved in this DNA replication process. So, you require the helicases, which actually going to separate the two different strands. Then you require the primase, which is actually a RNA polymerase. So, it is actually going to be required for making a synthesis of the RNA primers. Then you require the single stranded DNA proteins. So, single stranded DNA proteins are actually going to bind to the two strands like this, so that they should not come together and again re-anneals, and that is how it is actually going to maintain the single stranded DNA. Then you require the DNA polymerase, which is actually going to do a synthesis of new strand, and then you require the tethering protein, which is actually going to stabilize the polymerases onto the strands. So, DNA synthesis has the three steps. One is called as the initiation, which is actually going to prepare the DNA for the synthesis. So, what you are doing? You are actually taking a DNA and then you are actually generating the two strands. Okay. Then in the elongation steps, we are doing the DNA synthesis. So, once your two strands are ready, then what you can do is you are actually going to take and add the primers. So, in this case the RNA primers. So, both the primers are going to be sit, and then you are going to add the enzymes. So, the enzyme is going to be sit onto this strands, and that is how it is actually going to start the synthesis. And once the termination, so you can stop the DNA synthesis. So, what will happen? It will, once it will reach to this point, the enzyme is actually going to fall from the strands, and that is how it is actually going to complete the DNA synthesis, and it will enter into the terminations. So, now what you see is that these are the processes what we do under the DNA synthesis, and what happens inside the cell can be mimicked even outside the cell, because all these events can be controlled even by without going through the some of the enzymes what you require. For example, you require the helicases, so that it actually going to generate the single stranded DNA, and that is how it is actually going to make the and then you require the single stranded DNA binding proteins, so that the DNA strands what been generated by the helicases will remain as the single stranded. So, these two processes can be simply be done by if you heat these particular strands. So, if you heat this particular strand, what will happen is that the double stranded DNA is going to be open into the single stranded templates, and that is how you can be able to utilize them. Now, in the elongation phase, what you can do is you can simply add the small stretches of the DNA sequences, which is actually going to be called as the primer, and that is how it is, and then you can add the enzyme. So, that is how it is actually going to start the synthesis process, and once it reaches to the end, it is actually going to be fall from the template, and that is how it is actually going to end up into the terminations. So, keeping these events and realizing that these events can be done under the in vitro conditions, people have conceptualized the process of polymerase chain reactions, and that is how they actually started developing the technique. But there are multiple steps and multiple phases in which the polymerase chain reactions is being developed. So, there are multiple events what people have done even to develop the PCR. So, in 1950s people have discovered how the DNA replication is happening, and that is being done by the Arthur Kornberg, and he discovered the first DNA polymerase and other factors like helicases and primase. So, in 1950s, when the Arthur Kornberg actually discovered the DNA polymerases as well as the mechanism of DNA replications and helicases and primases, that is how the people know that the DNA is actually being replicated and it requires an enzyme, and there is an enzyme. But what is the problem? The problem is the DNA polymerase, what is being discovered by the Arthur Kornberg, is actually the temperature sensitive, which means it is cannot withstand a very high temperature. And you know that, as I said in the last slide itself, that when you want to mimic these conditions under the in vitro conditions, the only way you can be able to skip the helicases and all other proteins is that you can actually heat up the DNA, so that the DNA will come into the two strands, and those two strands will remain as a single stranded strands only if you keep the temperature very high. But once you bring the temperature high, the enzyme what is being reported by the Arthur Kornberg is actually going to be inactivated, and that is why nothing happened until in 1976 the people have discovered the first thermostable DNA polymerase from the Thermus acutus, and that DNA polymerase is called as the tag DNA polymerase, and that tag DNA polymerase is very stable. It can be withstand the temperature of 95 degrees Celsius, which means you can be able to use this enzyme on multiple rounds, and you do not need to you do not need to add the enzyme on for every cycle you have to continue. Then in 1983, Kary-Mullis synthesizes the DNA oligo probes for sickle cell anemia, and then 1983 he only did the repeated thermal cycling was first used to clone a small segment of genome DNA. And then in 1984, the Kary-Mullis and Tom White tried the design experiment to test PCR on the genomic DNA, but the amplified product was not visible in the agarose. Then in 1985, the first patent was filed for its application regarding the detection of the sickle cell anemia mutations, and the 1985 the first use of thermostable DNA polymerase in PCR was started. Out of only the two enzymes, the tag DNA polymerase known at the time, the tag was found more suitable for the PCR. And 1985 to 1987 people have discovered a lot about the different types of instruments, and so there are a lot of discoveries and development later on with the help of the instrumentations. So, what basically people are doing in a polymerase chain reaction? So, PCR is a repeated cycling reaction that involves the mechanism of DNA replications. It results in the production of multiple copies of DNA from a single one. The whole process involves three events. One is called denaturation, annealing, and the elongations. So, what happens? So, you can imagine that you started with a original copy of the double stranded DNA. So, under the denaturation, when you have heated the sample at 95 degrees Celsius, what will happen is that the two strands of the original copy are going to be separated, and it will give you the two strands. Now, what you are going to do is you are going to enter into the annealing phase, where you are actually going to lower down the temperature, so that the primer are going to bind, and then you are actually going to have the primers. So, it is actually going to add the primers, and that is how it is actually going to attach the primer on one end of this DNA as well as the one end of this DNA, and then the enzyme will enter into the elongation phase, and the enzyme is actually going to utilize this small primer DNA, and that is how it is actually going to synthesize the whole strand, and that is how you are actually going to get the two copy a single double stranded DNA at the end. So, this is actually is going to constitute the first cycle or the first cycle. After the first cycle, since you started with the one DNA, you are actually going to get two DNA, but as you can see, it is actually a semi conservative, which means the one strand is from the original DNA and the second strand is the newly synthesized DNA strand. Now, you can imagine that the same thing will happen in the second cycle, but now these one also is, these two strands are also going to serve as a template, and that is how you are actually going to get the 4 copies, and in the third cycle you are going to get the 8 copies, which means a DNA fragment of interest is used as a template from which a pair of primer or short oligonucleotide complementary to both the double strand of the DNA are made to prime the DNA synthesis, where the direction of synthesis or the extension is 5 prime to 3 prime, as it was in the case of DNA replications. The number of amplified DNA or the amplicon increases exponentially per cycle. For example, if you start with the 1 DNA molecule, after first cycle it is going to be 2 DNA, after second cycle it is going to be 4, after third cycle it is going to be 8. So, if you continue that, it is actually continuing like this cycle, like so. So, it is actually doubling after every cycle, and that is how it is actually going to give you very high amplification even from the one molecules. So, if you imagine that if I started with 1 microgram of DNA, it is actually going to be several microgram of DNA, and even within the 20-25 cycles. So, the amount of amplified DNA you can be able to calculate simply by putting this formula, that is the C is equal to C0 1 plus E to the power n, where the C is the final amount of DNA, C0 is the initial amount of DNA, E is the efficiency, and n is the number of cycle, and S is the slope of the expansion phase, and that you can be able to calculate from the this particular equations. So, you can see that the polymerase chain reaction actually utilizes the similar concept what is happening in the during the DNA replications, but instead of utilizing the multiple factors and such complicated mechanisms, what you are doing is you are simply heating the reactions, you are making the 2 strands, then you are adding the primers, which means the small DNA stretches, and then you are asking the enzyme to utilize these primer to synthesize, and then once the synthesis is over, then again you are bringing the temperature back and continuing the same cycle, and that is how you are actually making the multiple reactions. So, the polymerase chain reactions requires the 4 events. So, if you set up the polymerase chain reactions, you require the initial denaturations, which means you have to heat the PCR mixture at 94 to 96 degree for 10 minutes to completely denature the template DNA. So, when you are going to start the PCR reactions, you have to first bring the reactions, and then you are going to first do the initial denaturations, where you are actually going to heat up the mixture at 95 degrees Celsius for 5 to 10 minutes, and then you are actually going to enter into the stage 2, where you are going to again heat up the template for 30 seconds to 45 seconds, and that actually is going to be a denaturation steps. So, in the denaturation steps, this is the first step in which the double stranded DNA is going to be denatured to form the 2 single stranded DNA by heating the DNA at 95 degrees Celsius for 15 to 30 seconds. Now, you are going to lower down the temperature. So, that is going to be annealing steps. So, in the annealing step, this is the annealing step where the lower temperature like 50 to 65 degrees Celsius, the primers are allowed to bind to the template DNA, and annealing time is 15 to 30 seconds, and it depends on the length and the bases of the primers. So, what you are going to do is you have lowered down the temperature, so that the primers which are actually been floating and the which are small stretch of DNA are now go and bind to its complementary DNA present onto the template, and now the enzyme will recognize this particular the complex, and that is how the enzyme will start the synthesis. So, after the annealing, it will enter into the elongation phase. So, this is the synthesis step where the polymerase chain perform the synthesis of the new strand in the 5 prime to 3 prime directions using the primers and the DNTPs or the deoxynucleotide triphosphates. An average DNA polymerase adds about 1000 base pair per minute, which means after the annealing is over and the primer dimer has formed the complex, you are again going to increase the temperature, so that you will ensure that the template DNA should not bind to each other, and then the enzyme will come sit onto these complexes and it is actually going to do a DNA synthesis. And as an average, the 1000 base pair is going to be synthesized within the minute, when if you supply the adequate amount of the deoxynucleotide in the mixture. So, step 1 and 2, 3 will make one cycle. So, this stage 2 means like step 1, 2 and 3 is actually constituting the first one cycle, and you can ask the machine to continue this cycle for another 30 rounds, and that is how it is actually going to make lot of amplification. Once that lot amplification is over, then you can ask the things to enter into the next phase, and after the cycles are completed, the reaction is held at 70 to 74 degrees Celsius for several minutes to allow the final extension of the remaining DNA, and then you are actually going to ask to the final holds. So, once the stage 2 is over, where you can actually do a denaturation, annealing and elongations, and you can do for multiple rounds, then you can ask to enter into the stage 3, where you can just do another round of extension, which is called as the extended extensions for 10 minutes. So, that whatever the replicons or whatever the amplicons are there in the reactions and which are not complete by the end of this time, it is actually going to be completed in another 10 minutes, and then you can ask for a hold at 4 degree for infinite period of time. So, what are the things you require for doing the PCR? So, this is a classical example of one of the reaction cycles. Like you are going to do initial denaturations at 95 degrees Celsius for 5 minutes, and then you are going to have a thermal cycling, like these are the things are going to continue for the 30 cycles. And so, in the 30 cycle you are going to have the initial denaturations like the 95 degrees Celsius for 30 seconds, then you are going to have the annealing, which is going to be 55 degrees Celsius for 30 seconds. So, this is not fixed, this has to be decided based on the TM as well as the annealing temperature of the primer dimers and all that kind of parameters. And then you are going to have the extension. So, the extension will be at 72 degrees Celsius for another 45 seconds. So, extension time is also being calculated based on the size of the target DNA, because as you might have seen that the 1000 base pair is going to be synthesized in 1 minute, which means you can simply divide your target DNA by the 1000, and then you can put those many number of minutes. For example, if I am amplifying a DNA which is of 2 kb, then what I can do is I can simply give the extension time slightly above to the 2, because the 2000 is being divided by 1000, and that actually is going to give you the 2 minutes, but it should be slightly bigger to the 2 minutes. So, what I will do is I will give 2.1 or 2.2, so that there will be enough time for the enzyme to complete the extension process. Otherwise, there will be lot of amplicons are going to be there which are not being complete. And then you are going to have the final extension. So, final extension would be for 72 degrees Celsius for 5 minutes, and then there will be a final hold where you are actually going to ask the machine to keep the reactions at 4 degree, because the DNA is very stable at 4 degree, it cannot be remain stable at higher temperature. So, you can ask that the samples to be stored at 4 degree. There are requirements for the PCR. So, before you start the PCR, you should know all this, then only you can be able to do the PCR. What is these requirements? The first requirement is that you should know the DNA sequence of the target DNA, because if you do not know, then you will not be able to synthesize the primers, because this information is required to synthesize the primer, and the primers are actually being the complementary strands, complementary sequence to your target DNA. Then you require the thermostable DNA polymerase, for example, the tag DNA polymerase, which is not inactivated by the heating at 95 degrees Celsius, and then you require a thermal cycler, which is machine which has been used to program to carry out the heating and cooling of the sample over a number of cycles. And with this I would like to conclude my lecture here. Thank you.